The CRISPR-Cas9 system is a DNA editing tool that stands for Clustered Regularly Interspaced Short Palindromic Repeats and CRISPR-associated protein 9. First observed in bacteria, CRISPR-Cas9 is a means of defense against viruses. As foreign viral DNA enters a bacterium, it's processed into smaller fragments, which may be inserted into a region of the bacterial genome called a CRISPR locus. When the region is transcribed, the product associates with smaller RNAs called tracer RNAs, which may help to orient both the Cas9 protein and RNase to the molecule, the latter of which cleaves the transcript. The end result is several complexes, each consisting of a Cas9 protein, tracer RNA, and a CRISPR RNA, derived from DNA in the locus. The CRISPR RNA in these structures recognizes and guides Cas9 to viral DNA, which is then cleaved and destroyed. Scientists harness CRISPR-Cas9 by synthesizing individual RNA molecules that mimic tracer RNA and CRISPR RNA, which can target a gene of interest. For example, when two such guide RNAs are introduced into cells with Cas9 and both target the same gene, a sequence can be excised. Once this target region is removed, the cut ends are reconnected, and the effects on the cells are observed. Thus, the CRISPR-Cas9 system is modified from a bacterial mechanism and can be employed for an array of gene editing techniques.
Bacteria continuously face infections from bacteriophages, viruses that infect bacteria. To deal with this threat, bacteria have evolved a sophisticated adaptive immunity system known as the CRISPR Cas system to destroy bacteriophage DNA in case of reinfection. This system involves 3 different processes: incorporation of the bacteriophage DNA segment into the bacterial genome, production of the CRISPR RNA and the Cas protein, and CRISPR RNA Cas-mediated cleavage of the bacteriophage DNA. When a bacteriophage attacks, it attaches to the surface of the bacterial cell and inserts its DNA into the bacteria, which is then cleaved by the bacterial system. A short segment of the bacteriophage DNA is then added into specific regions of the bacterial genome called CRISPR, clustered regularly interspaced short palindromic repeats. These are genomic regions where bacterial sequence repeats are interspersed with the short varying spacer sequences from different bacteriophages. These spacer sequences serve as a memory for the bacteria of all the bacteriophages that have previously attacked. Bacteria use spacer sequences to rapidly identify and destroy DNA from a particular type of bacteriophage if it attacks again. The transcripts from the CRISPR region are processed to produce CRISPR RNA molecules, around 30 nucleotides long, that contain the spacer sequence and the nearby bacterial repeat sequence. The CRISPR-associated, or Cas systems, encode the Cas protein. This Cas protein then associates with the CRISPR RNA molecule to form a ribonucleoprotein complex. When the same type of bacteriophage attacks again, its DNA is recognized by its specific spacer sequence present in the CRISPR RNA. Associated Cas protein then, either alone or with the help of multiple proteins, cuts both strands of the bacteriophage DNA. The principles of the CRISPR-Cas system and its components can be used to knock down or modify any gene in an organism using its complementary guide RNA. Different types of the CRISPR-Cas system are present in bacteria and archaea. Among them, the CRISPR-Cas9 system is one of the most well-studied and widely used for different practical applications, as it can be easily and effectively reprogrammed to target different genes.